Hello, this is me, Dr. Tanya Hashik. Today's topic of discussion is spermiogenesis, structure of sperm, and axial filament. So the first topic of discussion is spermiogenesis. So what is spermiogenesis? We have already discussed about spermatogenesis. So spermiogenesis is the third stage of spermatogenesis, and spermiogenesis, it is a metamorphosis of spermatid to form spermatozoa, and here certain changes occur in the spermatid to form spermatozoa. So this is a spermatid and this is a spermatozoa, and in spermatid certain changes occur, and as a result spermatozoa is formed. So spermatid have so many structures, and all these structures are involved in the formation of a spermatozoa. So Golgi apparatus is involved in the formation of ectoderm. Mitochondria, it forms a sheath in the middle piece. Nucleus, it is involved in the formation of head. Proximal centriole, it is involved in the formation of neck, and distal centriole, it is involved in the formation of the middle piece, and thus each structure in the spermatid is involved in the formation of the spermatozoa. Now we will discuss this in details, that is the metamorphosis, how this occurs. So now we will discuss about the various events that takes place during the process of spermiogenesis. So the first one is nuclear condensation. That is, the nucleus undergo condensation and it moves towards one of the pole, and it results in the formation of head of spermatozoa. So the nucleus undergo condensation and it moves towards one of the pole, and it results in the formation of head of the spermatozoa, and that process is called as nuclear condensation. So this is the nucleus. It undergoes several changes and it results in the formation of head of the spermatozoa. So the second is the Golgi apparatus. This is the Golgi apparatus. It undergoes certain changes and it results in the formation of ectoderm cap, and it covers 2/3 of the nucleus. That is, this is the Golgi apparatus. It undergoes certain changes and it results in the formation of ectoderm cap, which covers 2/3 of the nucleus. Next is the proximal and distal centriole. So in a spermatid, proximal and distal centriole, they are seen together. When a spermatid is converting into a spermatozoa, the proximal centriole, it becomes spherical and it comes and lie in the neck portion of the spermatozoa, and the axial filament appears to come out of it. That is, the axial filament, it appears to come out of the proximal centriole. And the distal centriole, it becomes the distal end of the middle piece. It forms a ring in the distal end of the middle piece. So this is a spermatid, and these are the two centrioles, that is the proximal and the distal centriole. So here in this figure, in a spermatozoa, you can see that the proximal centriole, it becomes spherical and it comes to lie in the neck, and the distal one, it forms a ring in the distal end of the middle piece. So this is the distal end of the middle piece, and it forms a ring in the distal end of the middle piece called as annulus. So next is the mitochondria. So when a spermatid is converting into spermatozoa, the mitochondria, it forms a spiral sheath and it covers the middle piece, that is in between the neck and annulus. So the axial filament in this area is covered by the mitochondria. It forms a spiral sheath in the middle piece, and the axial filament in this area is covered by it. So the axial filament along with the spiral mitochondria form the middle piece of the spermatozoa. So this is the mitochondria. These two are the mitochondria, and these mitochondria, it come and forms a spiral sheath. So this is a spiral sheath. It forms a spiral sheath around the middle piece, and the axial filament, we have already learned that the axial filament appears to come out of the proximal centriole. So this axial filament will be covered by the mitochondria, that is the spiral sheath of mitochondria. So the axial filament along with the spiral sheath of mitochondria forms the middle piece. Next is the cytoplasm. The cytoplasm that is seen in the spermatid, most of it shed off during the conversion of spermatid to spermatozoa, and it shed as residual bodies, and they are engulfed by the Sertoli cells. So the most of the cytoplasm of the spermatid is engulfed by the Sertoli cells during the conversion of spermatid to the spermatozoa. And next is the cell membrane, and the cell membrane, it persists and it acts as a covering for the whole spermatozoa. So the cell membrane, it persists and it acts as a covering for the whole spermatozoa. So these are the events that takes place during the conversion of spermatid to the spermatozoa. So in this figure you can see various changes going on when a spermatid is converting into spermatozoa. In the stage 1 you can see several changes. This is the nucleus, the mitochondria, centriole, the Golgi apparatus. So in this stage you can see the axial filament is coming out of it. And here you can see the proximal and distal centriole. The axial filament is seen appears to come out of the proximal centriole, and the mitochondria is moving downwards. So here you can see the annular string, acrosome, cap is forming. So here again there is lot of change, and this is a proper spermatozoa. So here you can see every part of the spermatozoa, that is the head, the neck, the middle piece and the tail. So these are the stages when a spermatid is converting into a spermatozoa. So now we will discuss the structure of a spermatozoa. So when a spermatid is converting into a spermatozoa, it is immature, that is it is immobile, and further changes only takes place within the epididymis, and after that only a spermatozoa becomes a mature mobile spermatozoa. So now we will discuss the structure of a spermatozoa. So a spermatozoa has a head, a neck, a middle piece and a tail or principal piece. So now we will discuss the structure of head of a spermatozoa. So spermatozoa head, it is pyriform shape, it is 4 micrometer in length, and it is derived from the nucleus, and the head contains highly condensed chromosome, that is it contains 23 highly condensed chromosomes, and the chromatin is highly condensed and thus it makes stress resistant, and the head is covered by a cap called as acrosome. So this is the acrosome cap, and it contains enzyme that helps in penetration. So this is the head of a spermatozoa. It is pyriform in shape and it contains highly condensed 23 chromosomes, and the head is covered by a cap called acrosome, and it contains an enzyme and that is helpful in penetration. So the next part is the neck. It is a narrow area, it is a narrow region, and it contains basal body that is funnel shaped and also spherical centriole, and it connects that is the basal body, it connects the head with the other portion of the spermatozoa, that is the basal body. It has a convex articulating surface. It connects with the implantation fossa of the head and thus connects head with the other region of spermatozoa. So next is the neck portion. So it has a centriole and a basal body. Basal body is funnel shaped and it has a spherical centriole, and the basal body, it has a convex articulating structure which connect with the articulating fossa of the head, and thus it connects head with the other portion of the spermatozoa. So now we will discuss about the middle piece and the tail, and tail is also called as the principal piece. So this is the middle piece and this is the tail region. So the axial filament, it arrives just behind the centriole and it is seen throughout the middle piece and also in the tail. So the axial filament in the middle piece, it is covered by a sheath of mitochondria. It is covered by a spiral sheath of mitochondria. So this is the spiral sheath of mitochondria. So the portion where the middle piece joins with the tail, the axial filament passes through a ring called as annulus. So this is the ring, and the portion where the middle piece joins with the tail, the axial filament passes through a ring called as the annulus. And next portion is the tail or the principal piece, and it contains only the axial filament and it lacks mitochondria. So this is the structure of a spermatozoa. It has got head, neck, middle piece and also a tail. So now we will discuss the structure of axial filament. This is the transverse section. Here you can see in the center a pair of central fibrils which is surrounded by 9 pair of fibrils, and the 9 pair of fibrils is called as the doublet. So a pair of central fibril which is surrounded by 9 pair of fibrils, and that is called as a doublet. And outside the doublet you can see petal shaped fibril each. That is 9 petal shaped fibril can be seen, and that is called as the coarse petal. And all these fibrils are kept in position by series of coverings. So all these fibrils are kept in position by means of series of coverings. And the outside fibrils, there is a fibrous sheath. So this is the fibrous sheath outside the covering, outside the fibrils. So in case of middle piece, you can see that the axial filament is covered by spiral sheath of mitochondria. So in case of middle piece, the axial filament, it is covered by the mitochondria, and the whole spermatozoa, it is covered by the plasma membrane. So the outside the fibrous sheath is a covering of plasma membrane. So this is the end of today's class, and for more videos, please subscribe to my channel and press the bell button.